Hello class and welcome to our fifth lecture video here for Unit 6. In this video we're going to discuss the process of DNA replication, which is also referred to as semi-conservative. So what do I mean by that? If we just look at this diagram here on this page, let's start off here with a single DNA strand. What's going to happen is it's going to slowly unwind, and each side of DNA is going to be used as a template strand to make two brand new double helix molecules that you can see going on until you get to two identical daughter double helixes. So what semi-conservative really means is that you only need one side of DNA to make a brand new identical double helix that is similar to the original strand that you made. All right, fully conservative would mean that you would actually have to use both sides of DNA, both the original sides, to get one brand new double helix, but that's not the case. So moving forward here, just to get through some of the basics for DNA replication. Obviously, for the definition, you don't have to really focus on the complex specific definition here. It's the process of copying DNA or chromosomes, period. And the way it's going to be done is, as I just mentioned, via this semi-conservative model. Again, you only need, when you unzip DNA, you only need one side of it, because if you have an A on one side of DNA, we should all know by this point the other side should have been T. So you would just add a T. Or if one strand of DNA has a G for guanine, you know cytosine C had to be on the other side. So we only need one side of DNA, and we call that semi-conservative. Location, for all eukaryotes, like you, me, plants, fungus, it's going to be within the nucleus. And when is this process occurring? It is occurring during the S phase of the cell cycle. So recall in G1, a cell is growing, but eventually it gets too big where it passes the restriction point. As the cell gets too large, its rate of diffusion begins to suffer, decreases. So the cell, if it can, will enter the S phase where it copies all of its DNA, or in our instance for humans, all 46 chromosomes would be duplicated now during the S phase, before we eventually begin and enter cell division, which is going to be mitosis. And we'll cover that after we're done here with DNA replication. So any process that we've done this year, we really only looked at it being done by one enzyme. But with DNA replication, all right, on the next couple of slides, we're actually going to see that there is at least 4 specific enzymes that we're going to take into account that are involved with the process of DNA replication. They're going to be helicase, DNA polymerase, what I'm going to call RNA primase, and then finally DNA ligase. So let's begin. I've broken down DNA replication into 3 key steps. Now I could break it down into 100 if I wanted to, but I'm going to try to simplify it as much as I can. Step 1 is definitely the easiest. For step 1, pretty much we're going to use an enzyme called helicase that will both unwind and straighten a double helix, but then it's going to separate it into 2 template strands by breaking, here's the key that you need to remember, the hydrogen bonds between the nitrogen bases. So it looks like this down below. All right, the enzyme helicase will bind onto DNA. All right, and we'll wait till it restarts over here, and it just starts to unwind and break the hydrogen bonds between the 2 template strands. So now I have one side of DNA up here on the top, and then one down here on the bottom, and we can use each side as a template to produce a brand new double helix. As helicase unwinds the DNA, sometimes they call this a replication fork, because it's like a fork in the road. And also notice that over here it describes the top strand being what we call the leading strand, and the bottom the lagging. Because as we're going to see, the leading strand gets made much quicker as opposed to what the lagging strand is going to be made down the line. And it all has to do with the orientation of the sugars. Remember, you have a 3' and a 5' side of DNA. What we're going to see is the 3' side is made much quicker than the 5' side is. So for step 2, so this slide here is going to be a little bit more technical. We're going to use an enzyme next called DNA polymerase. All right, and it's going to want to bind to each of the 2 template strands, both the top and the bottom. Its job is to read each template strand, read the nucleotide of each template strand, and add a corresponding or complementary nitrogen base. So if DNA polymerase reads an A on one strand, it's going to make sure that it adds a nucleotide that contains thymine, and so forth. The trick is this, though. DNA polymerase can only make new DNA or add nucleotides in one specific direction, the 5' to 3' direction. So that's the 5', remember again, that means the 5th carbon on deoxyribose is facing one direction. And you want to make sure then the 3rd carbon is facing in the opposite direction. So no matter if it's a leading or lagging strand, DNA polymerase can only add new bases or nucleotides in the 5' to 3' direction. So you have your two template strands you just unzipped. The one that is considered the 3' side will have an OH group. DNA polymerase can only bind to a template strand is if there is an OH group available, and that only is on immediately the 3' end of DNA. So the 3' end of DNA will allow DNA polymerase to immediately bind to it. DNA polymerase will then start to add complementary nucleotides in the 5' to 3' direction continuously towards the replication fork, that little fork in the road where helicase was splitting the DNA. Since this happens continuously, we're going to call this strand the leading strand because it's always in the lead and how quickly the new double helix is being produced. So make sure you look at this diagram accurately on the next page as I start to review this. So here is my original double helix. Here is helicase that's moving towards our left, unzipping the DNA into two template strands. So the original strands over here would be the dark blue. Now, if I follow my laser pointer over here to the edge of the original 3' direction, and we're going to call this the leading strand, the 3' end, as I mentioned on the last slide, has an OH group. That OH group allows, in this color over here, DNA polymerase to bind. And DNA polymerase, again, can only make DNA in the 5' to 3' direction here where my laser pointer is accentuating. So when DNA polymerase binds here, it starts to make the new strand of DNA, which is the lighter blue, in the 5, follow the arrows, to 3' direction right here. And see how it's being made non-stop and continuous? Well, that just happens to be, the 3' direction happens to be the same direction that helicase is moving in. Again, here is a little blurb here where you can see where the actual bases and sugars here look like in DNA. See this OH group on the 3' side? So that's what was available right here. Polymerase bound to it and has been synthesizing DNA continuously in the same direction as helicase is moving. Now on the other strand of DNA down here in the bottom, this original dark blue strand is the 5' end. Well, on the 5' end, there's no OH group, so DNA polymerase is going to have some difficulty binding to it. But then again, DNA polymerase can only make DNA in the 5' to 3' direction, so it's going to have to go not from right to left like you see up here continuously. It's going to have to move from left to right, which means it's always going to be waiting for helicase to move down to open up a new section of DNA, which is why it's going to take longer or it's going to lag behind. So if I just move on to step 2 continued, where we can discuss the lagging strand, again, it's in any picture, it's always going to be the bottom strand, the 5' end, that we call the lagging, like you can see down here in this picture. So a couple new things have to happen here first. We need to add an OH group down here to the lagging strand, and that's going to be done by an enzyme called RNA primase. RNA primase will add an OH group, which now allows DNA polymerase to bind to it and make a little short segment of DNA called an Okazaki fragment, named after Dr. Okazaki, who discovered this process of semi-conservative replication, which won him the Nobel Prize. So down here in the picture, you see how these little short blue arrows? Those are the Okazaki fragments. You got to make DNA in the 5' to 3' direction, which is the other way this time. So you're always waiting for the fork to open up. You're always waiting for helicase to move down a little further. So if I move forward here to this diagram, RNA primase right here in pink adds a little primer, which is in red. Notice what's on the primer? A 3' N, which is going to have an OH group on it. That now allows DNA polymerase to bind to the primer and synthesize an Okazaki fragment you can see here in the 5' to 3' direction, which is going to be left to right. But these fragments are always going to be in between these red primers that have to get laid down every time helicase moves down further. So you're going to have these discontinuous fragments, these Okazaki fragments. Now what happens is, eventually, once all these primers are removed and you add in, instead of RNA bases that are here, you add some DNA bases, you're going to have these little missing gaps. You can see here where my laser pointer is, and this black circle here is accentuating. So what that really looks like is, so right here, number 1 light blue is an Okazaki fragment, and over here in light blue, number 2 is an Okazaki fragment. You want to connect those together. You don't want to have a missing gap in your DNA. So what happens is, you can see right here, I've blown it up, there's the gap, and it's really just a phosphate group that's missing. So we're going to have a last enzyme come in here called DNA ligase, which is going to connect the 2 Okazaki fragments together. So down here would be your original strand of DNA, which is already connected, but up here would be number 1 and number 2 Okazaki fragment, number 1 right here, number 2 right here. DNA ligase finally comes in and connects these 2 Okazaki fragments together by adding a phosphate group, and now you complete the full identical double helix molecule. So to finish out our notes, and I just described this, again, the Okazaki fragments on the lagging strand will be connected together by a phosphate group. And who does that? DNA ligase. Our end result from semi-conservative replication should be 2 identical double helix DNA strands. All right, and you can see that over here in the diagram. All right, each one should be identical to what the original strand was, but each brand new strand will be 1/2 original, pink strand, and 1/2 brand new, blue, which we're going to call complementary. So again, that's semi-conservative replication. Each strand only needed to use 1/2 of the original DNA. If I proceed forward here, all right, just another slide or two, I can show you this in action to finish up. So here is helicase. From this perspective, it happens to be moving to our right, but helicase right here is unwinding the DNA and separating it by breaking the hydrogen bond. So here's our fork. Up top is our 3' N, which has an OH group, which allows right here DNA polymerase to bind to it. And you see how it's synthesizing DNA continuously, just heading towards the fork down here? It's because it's making DNA in the 5' to 3' direction. But down here, the lagging strand, DNA has to be made in the opposite direction for it to be 5' to 3. So to do that, here's primase. It adds a little primer that would have an OH group on it, allowing DNA polymerase here to bind and make an Okazaki fragment. You can see this little fragment right here. Eventually, this little primer is removed, and then it's sealed by DNA ligase. So if I let it proceed through one more time, I want you to look right here. There's going to be a little missing gap once that primer is removed. And the gap between this Okazaki and this Okazaki, there it is, you can see the gap, is going to be sealed eventually by DNA ligase. So leading is continuous, and you can see why. 5' to 3' direction is nonstop. But discontinuous here on the bottom lagging strand, because you're always waiting for helicase to move down before you can add another primer and make another Okazaki fragment. Again, try your best to memorize the function of the 4 enzymes and realize really what the difference is between the leading strand having that 3' N and the lagging strand having the 5'. So that means it has to be made in the opposite direction.